Replication of protein and DNA through the formation of double helix structure. Early processes of replicating protein and DNA. The mechanism that first produced proteins and DNA. Formation of a double helix is formed by pairing of hetero-chiral molecules together with linkage of homochiral molecules. Amino acids of the same chirality are peptide bonded to synthesize proteins. If the chirality of adjacent molecules is different, they are in contact with each other in a mirror image, so they cannot be connected continuously. When the enantiomers of front and back sides face each other as shown in the upper left corner of Fig. 1, and intersects by reverse progress for the matching, the overlapping areas are face-to-face by bumpy relationship. A double helix structure is formed when the racemes of amino acids are elongated by homochiral bonds. The double helix amino acids will form a protein molecule. Like amino acids, nucleic acids are asymmetric molecules, so RNA and DNA can also form double helix structures. How double helix structure was first produced. Formation of double helix structured proteins by hetero-chiral pairs and homopeptide bonds of amino acid molecules. When hetero-chiral molecules proceed in crossing and the right side of left protein is matched with the left side of right protein as shown in Fig. 2, a double helix is formed in the paired area. L protein in double helix plays a role of a mold of D protein, and D protein plays a role of a mold of L protein. According to Wallach's rule, racemic crystals, which are composed of pairs of hetero-chiral molecules, are denser than L or D type crystals. How was DNA formed? Double helix of DNA is produced by an amino acid unit with crossing over. Asymmetric molecules such as amino acids are enantiomers that cannot be superimposed unless the bonds between molecules are broken and reconnected. The formation of its enantiomers builds a double helix structure. Fig. 3 shows Watson and Crick DNA model. The amino acid unit on the formation of a double helix for different chirality is the same size and the matching process of replication is carried out by the segmentation of amino acid unit. When the hetero-chiral pairs are linked in series, a double helix structure will be formed. The replication of the double helix is performed by matching each amino acid in the order of the sequence of amino acid units. When the generation of pairs by racemization is done randomly, it is not possible to replicate the exact long protein. How to replicate a double helix of DNA? DNA is replicated by mechanism of racemization. When DNA helicase unwinds the double helix structure of DNA, two hetero-chiral single strands are produced. As shown in Fig. 4, separated leading strand and ragging strand are added to replicate a double helix structure of DNA. The DNA is replicated by the effect of a racemization. The direction of unwinding the double helix structure and the preceding of the leading strand are the same. However, the preceding of production of lagging strand is opposite. Formation of Okazaki fragments. A new strand begins to be produced is set by the DNA primer as shown in Fig. 4. Okazaki fragment will form a double helix as shown in Fig. 4. The complementary base pairs of Okazaki fragment continue to create a double helix structure, which elongates in the opposite direction until it reaches the double-stranded region. Then, formation of next Okazaki fragment is started from the point of latest shingle strand. How to store the molecular sequence of amino acids. How to connect amino acid molecules to nucleic acid genetic codes. Since the RNA that performs the task of protein replication have a short lifespan, there is a need for a mechanism to store the molecular sequences of amino acids. The information of replication is stored by nucleic acid molecules of DNA and preserves them for a long time. Since replication of protein is carried out by amino acids and nucleic acids, both the mold of amino acid and the mold of nucleic acid have to exist at the same time in a cell. The aminoacyl tRNA that carries amino acids has formed from the molecular organization that connects the mold of amino acids with the mold of nucleic acids. Molecular structure of DNA. The base pairs of double helix structure of DNA are formed by hydrogen bonds. The DNA molecule is made up of nucleotides formed by carbohydrates and phosphoric acids formed by a combination of two bases, adenine A and thymine T and guanine G and cytosine C. The nucleotides bind to 5 carbon sugars and phosphoric acids, forming long chains, and 2 strands of DNA are linked by complementary base paired hydrogen bonds. The base pairs of RNA become racemic pairs, and the RNA in the racemic pair can replicate in a way that one of the templates is the other. Note that strands of the same chirality are connected from the 5' end to the 3' end, so the elongation of strands with complementary base pairs intersect. Racemic coupling between mRNA and tRNA. Nucleobases of genetic codes are set by amino acid units. In the mixture of the molecular groups of amino acids that make up proteins and the nucleotides that make up the genetic code, the hetero-chiral molecules form double helix and it enable replication by the mold method. On the other hand, homochiral molecules have the effect of linking with each other. The results of a lot of trial and errors, mRNA is produced by L-type amino acids and L-type nucleotides with a similar double helix structure, and tRNA is formed by D-type nucleotides and D-type amino acids. A tentative model that easily represents the correspondence of the atomic sequence of amino acid and genetic code of nucleic acids is shown in Fig. 7. The mRNA is used as a template for the amino acid molecular sequence, and the protein molecule is replicated by tRNA with the corresponding amino acid unit molecule. When RNA is produced from DNA, the oxygen of deoxyribose in DNA is removed, and it turns ribose of RNA, and thymine in DNA is converted to uracil in RNA. How an amino acid linked with the genetic code. tRNA was created instead of the tissue of molecules that act as synthase. The combination of amino acid molecule and genetically encoded nucleic acids depends on the type of amino acid. That is, there are 20 types of synthases. The tissue of those 20 molecules were replaced by 20 types of tRNAs. How to produce amino-acyl tRNA is shown in Fig. 8. At first, an enzyme complex is activated by ATP and is formed an amino-acyl AMP. The amino-acyl AMP carries an amino acid molecule. How to replicate protein in ribosome. Amino-acyl tRNA carries molecules of amino acids to ribosomes and replicates proteins. The organization of the molecule shown on the left and right outside of Fig. 9 is tRNA. The amino-acyl tRNA is the structure of the molecule with the amino acids. Amino-acyl tRNA is used to matching with mRNA in ribosomes, and the fitted amino acid of tRNA binds to a new protein. Conclusion. There are two types of molecules in amino acids and sugars, left-handed L-type and right-handed D-type. In living organisms, proteins are made up of L-typed amino acids, and nucleic acid sugars are made up of D-type. Until now, there has been no convincing explanation for the fact that living organisms use L-type amino acid molecules and D-type sugars. The chirality of related amino acids and nucleic acids plays an important role in the initial mechanism of protein replication was proposed in this video. When you cross your hands from your fingertips with the back side of your left hand contacting front side of your right hand, you can create a structure in which one side acts as a mold of the others. Therefore, in a state where the amino acid molecules that make proteins and the nucleic acids that make up DNA are mixed, the double helix structure is formed by the homochiral peptide bonding of the hetero-chiral amino acid molecules paired under the effect of racemic formation. After some evolutions, the protein was replicated by adding matched amino acids when the base of tRNA with amino acids is linked with that of mRNA. Thank you very much for your attention.